the effect of decomposing fish in prolonging red tide events. This will be presented by Dr. Mike Parsons from Florida Gulf Coast University. Next slide. Purpose is to provide an overview of recent research to assess the effect of decomposing fish in prolonging red tide events and evaluating fish removal mitigation strategies. The project is part of a state-funded Florida Red Tide Mitigation and Technology Development Initiative. The relation to the CCMP is that this ties to our water quality improvement action number 5 to reduce harmful algal blooms, specifically activity 5.1 to support the HAB research and monitoring and measures to reduce their environmental, social, and economic impacts, their identification and reduction of anthropogenic influences. I'll now turn this over to you, Dr. Parsons. Thank you for joining us this morning.
Sure, thank you very much. Everybody can hear me okay?
Yes.
Okay, great. Excuse me. So first of all, I'd like to acknowledge my other principal investigators, including Cindy Heil at Mote Marine Lab, Puspa Adhikari, Tim Allen, and Shelton Weeks here at Florida Gulf Coast University. Tim and Shelton in particular worked up the economic data that I'll be sharing with you. And this project was part of the Florida Red Tide Mitigation and Technology Development Initiative. Next slide, please. So basically what we're looking at is we're answering this question here on the bottom. Is fish removal and composting a viable option to mitigate Corynea brevis blooms? So basically you can see this feedback loop shown on the slide here, where when Corynea brevis starts blooming and cell concentrations get up into the hundreds of thousands of cells per liter of water, it starts producing enough brevitoxin that can get into the water or into the fish gills directly as the cells lyse when the fish are swimming through the water, and it will start killing fish. These fish will decay and they'll release nutrients, and those nutrients in turn can continue to fuel the red tide, which can then produce more toxin, kill more fish, producing more toxins. So the idea is, well, what if we remove these dead fish? Can we remove an important nutrient source and thereby offer some mitigation against the red tide? And then the second part here is, what do we do with those dead fish? So we're looking at composting and possibly using this as a fertilizer. Next slide, please. So what our objectives were really to, I'm going to move my little window here out of the way. We really wanted to get some better numbers on what the nutrient production of decaying fish was, so we can really get a better grasp on what we're dealing with here. We conducted a cost-benefit analysis of fish removal, and also we looked at composting once again. The idea would be to convert these dead fish into a fertilizer that could be used locally. Next slide. So we conducted a series of fish decay experiments. Basically, we took a moderately sized mullet and we had replicates. So we had around 20 mullet, I guess, each in one of these blue bins. And then we would just add seawater of 2 salinity. So we wanted to basically replicate full strength salinity in the Gulf of Mexico, and then more of a brackish condition of about 24 salinity that you would see in bays and estuaries. And then we would just incubate the fish and allow them to decay, and we would be measuring the nutrients as these fish decayed. So you can see 3 of our graduate students here working on the project, and this was the summer trial, and as you can imagine, working with decaying fish in the hot sun was a lot of fun for the students, so they really appreciated this project. We also ran the project in the winter months with mullet, to try to see if there was a temperature effect, which we expected in terms of how quickly the fish decayed and released nutrients. Next slide. So just in brief summary, what we found was ammonium was the main nitrogen-based inorganic nutrient that was produced by the decaying fish. We would expect that as the fish tissue would decay, ammonium would be the expected byproduct here. The numbers were up to about 50% higher than previous estimates, but phosphate was actually a bit lower, quite a bit lower than previously estimated. So what we're looking at here is we used mullet, previous studies used pinfish, and so it looks like we're going to have some species-specific variations in terms of nutrient releases. So we need to probably need to continue to get some good numbers on here so we can really estimate what that nutrient source is with the fish kill. That'll help basically drive that cost benefit analysis and, you know, our future actions on this. There really wasn't much of a salinity effect on nutrient release rates. So we would basically be treating a fish kill similarly if it was out in the Gulf of Mexico or up in the bays. We wouldn't necessarily have to operate any differently in terms of a timeline. But we did see a temperature effect. So during the summer months, as you'd expect, the nutrient release was greater. And so you might want to move more quickly in the summer months versus the winter months. And typically, we saw the nutrients would stabilize within days. And so if you want to try to remove the fish at the most opportune time, it'd probably be within a day or so of identifying the fish kill, because it's going to start releasing nutrients, you know, after about 48 hours, and it's going to be doing it at a pretty good pace up to a week later. So you have a short window to really act on it. But, you know, if you can't get to it for a week, you probably aren't doing any more damage. So we're starting to get some numbers here in terms of what the action plan would be if we decided that removing these fish while they're floating around in the water would be a good action to take. Next slide. So the second part of the project, we were looking at the economics of the situation. And so the first thing I want you to look at is that bottom number on the very bottom, which is about almost $12 million. And this is for Collier County and Lee County. And this was the estimated tourist tax revenue losses that were experienced in the counties in 2018 and 2019. So basically, we're looking at about $12 million per month while red tide was really prevalent. And by prevalent, we define that as greater than 100,000 cells per liter, which is typically the concentrations needed to cause these fish kills then. So, you know, just a quick, simple mathematical relationship. If we can keep fish kill cleanups below $12 million, then maybe we could at least break even if that fish kill removal would in fact reduce the red tide. And that's where the nutrient budgets come in and looking at these numbers. But, you know, another way to look at this as well is we are seeing significant losses in terms of red tide and the economy. And there's many different studies. I know many of you are familiar with different numbers and different ways to look at it. One thing that I think is interesting is the data really are not easy to come by, and the analysis takes a bit of effort. So there's different ways to look at this, and the more ways we can basically come up with these numbers, I think we'll get a better handle on the true economic impacts of red tide that really will drive these cost benefit analyses. Next slide, please. So we did have data from different counties in terms of cleaning up dead fish. Some of the dead fish would be floating in the water, others would be along the beaches. And so you can see that basically it's anywhere from $1,600 to $6,000 per ton of fish in terms of the cleanup material. So once again, if we're looking at $6 million per month in terms of lost revenue, and we're looking at up to $6,000 per ton of fish to remove it, we at least have some numbers that we can work with. And it looks like it is economically feasible to take these actions and remove the fish. But again, will that actually make a difference? Will that mitigate and reduce the red tide intensity? So we have a lot of the numbers in place here. I think the next steps here really are to take those nutrient release numbers. We need to get some better estimates on what, quote unquote, the average fish kill looks like in terms of number of fish and weights of fish. So we're trying to get better numbers on that. And then we can kind of start plugging in the numbers into a nutrient budget for Karenia brevis, and then really get a sense of, you know, where should we pull the trigger? How big does a fish kill need to be to take action on it? And as you all can imagine, you know, it depends on where the fish kill is, how spread apart the fish are, so on and so forth. But I think we have some good numbers that we can move forward with to continue this feasibility study. The next step then is, what do we do with all these dead fish? Right now, we basically just throw them into a landfill. can we do something else? Can we actually turn this into a positive product? Next slide, please. So we worked with a company called Wilco, and they have a product called SEB, which is a treatment. It's basically a composting accelerator, and they developed it for the livestock industry. And so what we show here are two 750-gallon tanks where we had a control that just had fish and water in it. It was just fresh water. And then in the middle photo here, you can see we have fish, and we added the SEB, which is basically a bacterial accelerant with an added carbon source. And what you may notice in the middle bin here, and on the far right, you can kind of see some pieces of seagrass and seaweed. These actually came from fish kill that was cleaned up off of Clearwater Beach. And so we collected about 500 pounds of fish, put 250 pounds in each of the left tank and the middle tank, and then we just basically let them degrade and compost. And on the left tank, you can see when we didn't add anything, most of the fish carcasses basically sank down to the bottom. There were some insect larvae acting on the fish tissue itself, but basically it just kind of liquefied and you had bones. If you look in the middle panel, it was more of a controlled process where the fish would actually almost firm up and even solidify, almost like a jerky material. And then eventually it would turn into this paste that you can see on the far right. So we could actually better control the composting process. This was a liquid composting process too. So it would basically create a liquid or semi-liquid fertilizer product. So we took that brownish material on the right, and then we ran NPK analyses on it, which is standard for fertilizer. And you can see it was nitrogen rich. It was a bit low in phosphate and potash. So if we did use this as a fertilizer, we would probably have to supplement it with phosphate or potash. One of the interesting things about this is potentially this could be an organic fertilizer, because these are natural wild fish. And so by removing the fish, you know, it's basically a natural product. And also this nitrogen source came from the ocean. So if you used it as a fertilizer and the fertilizer or the nitrogen got back into the ocean, you're basically just returning it from whence it came. So it could almost be a form of nitrogen neutral fertilizer. So there may be some environmental benefits here in terms of being nitrogen neutral, as well as being potentially an organic, at least a natural fertilizer. Next slide, please. But the big question is, the fish died because they were exposed to brevitoxin, the red tide toxin. And you can see it up there. PBTX is the symbol for the brevitoxin. It was back in the days when Crenia brevis was called Tychodiscus brevis. So it was a P and a B for the old genus and species name. You can see at the beginning of the composting experiment on August 2nd, both the control and the treatment showed the presence of PBTX3, which is one of the forms or one of the structures of brevitoxin. Brevitoxin has several different structures. They just slightly differ in chemical structure. We only detected three, Brevitoxin 3, but you can see after about 20 days, it actually disappeared in both the control and the treatment. So what this showed us is composting can remove the brevitoxin, and it really didn't require that SEB treatment. It's just the natural bacterial processes could break down the toxin. So this is promising as well. So this shows us that we can compost the fish, we can create at least a nitrogen-rich product, and it looks like that it'll be free of toxins as well. So we did a liquid composting process. There's probably just using a solid composting, maybe adding some more carbon sources, including, wood chips or other plant-based matter. I am not a composter. I know there's a lot of composting groups out there, anything from just homeowners to small companies that are really interested in this. And so I think the next step on the composting process would be working with these huge amounts of dead fish and trying to come up with the best composting product that could be used as a local fertilizer. Next slide, please. So in summary, ammonium is really the major source of nutrients, and Karenia brevis does really react to the presence of ammonium in the water. And it looks like removing fish may be a good mitigation strategy. We need to continue the process of what would a scale up look like, and when would removing fish be economically feasible and actually show, or we would predict a red tide reduction by removing those fish. It looks like composting may be a good end product of these dead fish. And the next steps really would be looking better in the field and at some of these large scale composting processes to see, are we on the right steps here? Does this look like a promising strategy? Next slide, please. So we're basically preparing for the next steps. We're looking for the next phase of the red tide mitigation program to see what the next funding cycle is. We'll be talking with some other composting groups, looking at talking with some of the counties and their event response and their fish kill responses to see if we can maybe do some pilot studies on removing the fish and looking at what nutrients and red tide do when we remove the fish. Of course, there'll be some controls needed and a bit of experimental design, and that'll probably be fish kill specific. So there's our work cut out for sure. This is my contact information here if anybody has any other questions, and that wraps it up for now. I'll be happy to take any questions if there's time.
Thank you so much, Dr. Parsons. I'll open this up for questions. If anybody has any, please come off mute.
I had a question, Doctor. Were you able to, have you spoken with Manatee County with maybe Dr. Hopes or Charlie Hunsicker about this past summer with what we did there?
I did not, no.
Okay. So we were kind of in a position where we're a brand new board, or at least a brand new majority, and kind of conservative Republicans. So the press already hated our guts. And then, of course, here comes the Piney Point, and then here comes red tide. And so we made it real clear that we were doing whatever the heck we could. So he took, if I understand it correctly, we had drones and then local fishermen hired to go. They would spot the fish kills out, offshore, and then go get it basically right away. And I don't know what the number are on how many tons we removed or things like that. But it was seemed pretty proactive and I think effective from what I was told. So I just wanted to just put that on your radar.
Sure. Yeah, because that's exactly what we're looking at. And then it'd be interesting to see what data were collected along with that. But what would be, I think, our next step in that case would be looking at red tide, looking at the nutrients, and see, hopefully in both cases, they would be reduced as these fish are being removed. But yeah, that's exactly correct. I'll definitely like to follow up on that. And so who can, I just see JS, so I'm not sure who's speaking, but who are the names again?
Sorry about that. I don't know how to change my whatever. This is James Satcher. So I'm a commissioner in Manatee County. Okay. And so our county administrator is Scott Hopes, Dr. Scott Hopes, Hopes. And that's probably the best place to start. Okay. And he can steer you in the right direction from there, I would think. And feel free to send me an email as well. That'd be great. We'll get you to the right people. I don't know, it was kind of just all hands on deck. Whatever you can figure out that might help, we're doing it. Oh, and so I don't know if they were gathering that much, data as we go or not. But it'll be interesting to talk to the different county response, and just see what the responses were, what they thought looked promising, what was a dead end, and just, kind of get a lot of the different coastal counties together and be like, hey, this is what worked in the past. This is what didn't. What's, can we have an action plan, like a hurricane action plan? Can we have a fish kill action plan where we just rip open the envelope, pull it out? Here's what we do. So I think that'd be great. Yeah.
I'm going to jump in
Absolutely.
and just share our next presentation, I think is strategically placed. And so I think we'll be hearing more about the fish cleanup that's being discussed.
Yep.
One
Any more
quick question. I missed a little bit of, you were saying the difference between getting the fish while they're still floating versus the fish while they've already come to shore. Could you kind of repeat that and expand on that just a little bit? Did you say it's much better to do it before they hit shore?
Not necessarily. I think the
Okay.
key is to basically intercept them as soon after death as possible.
Okay.
And so because they will accelerate their nutrient release up to about a week. So if they're left floating in the water for a week, that's bad. If you can get them
Okay.
within a couple days, that's good. And so when they would wash up on the beach during that, I mean, if they wash up on the beach within a day or two of dying, clean them up off the beach, but don't let them sit in the water for a week or two and then have them wash up on the beach.
Okay. I appreciate it. Thank you.
Sure. You're welcome.
Probably got time for another question or two. Dr. Parsons, I have a question just on the liquid composting process. What are the odors associated with that? I mean, in terms of where this can be done, does that need to be removed far away from communities, or is it something inherent in the liquid composting process where odor isn't a significant issue?
Surprisingly, odor is not an issue at all.
Wow.
And you know, we even had a colleague who had a bunch of chopped up fish in a 5-gallon bucket that he kept just in his office in his building. He's like, nobody even knew. And I said, I don't know about that, but you know, we ran the controls, which had nothing in them, and those were just horrible. And they were nearby, so you know, we still were dealing with the smell of, you know, rotting fish. But no, the composting, yeah, it was a very clean process. And my understanding is composting in general, when done correctly, is not a smelly process. I'm not a composter, so I can't guarantee that, but
But it sounds like that additive that was used contributed to less odor than
correct.
the control, which was just the fish dumped in a
Yeah,
barrel.
and not fun on that side of it.
Well, again, thank you for joining this morning. Thank you for the presentation and dialogue. Greatly appreciated.